Hey guys, welcome to the new video. Today I'm gonna go over chapter 5 of microbiology. Chapter 5 covers microbial metabolism, and yeah, let's just get into it. Okay. So, basic chemical reactions underlying metabolism. So, what is metabolism? Well, metabolism is the collection of controlled biochemical reactions that take place within a microbe. So, in other words, it is all of the chemical reactions in an organism. And there's two types. It is catabolism and anabolism. So, metabolism, ultimate function is to reproduce the organism. The metabolic process provides the necessary energy and materials for cells to function, grow, repair, and divide. So catabolism and anabolism are the two major classes of metabolic reactions. We first have catabolic pathways. Catabolic pathways break larger molecules into smaller products, and as a result, they release energy. And when they release energy, that's what you call an exergonic. For anabolic pathways, they synthesize large molecules from the smaller products of catabolism. So in other words, they build larger molecules from smaller components. Therefore, they require more energy than they release. And when they require more energy than they release, that's what you call an endergonic. Okay. So this image here shows the relationship between catabolism and anabolism as a part of cellular metabolism. So let's go step by step. So catabolism. What it does is that it breaks larger molecules, like nutrients, into smaller molecules called precursor molecules. As larger molecules are broken down, energy is released. And some of this energy is used to convert ADP, which is adenosine diphosphate, into ATP, which is adenosine triphosphate, which is the cell's main energy currency. Not all of the energy from catabolism is captured in ATP. Some of it is lost as heat. Um, the ATP stores energy for the cell, ready to be used for processes requiring energy like anabolic reactions. And so then we move into anabolism. Okay. So anabolic pathways use energy from ATP to build larger complex molecules like macromolecules from smaller building blocks, which is the precursor molecules right here. The ATP generated from catabolism provides the energy necessary for anabolism. ATP is broken down into ADP during these reactions, releasing energy. Uh, Anabolism creates larger molecules like macromolecules, which are essential for building cellular structure, such as the membranes and ribosomes. These macromolecules are also important for cellular functions like cell growth and division. So, in summary, catabolism is the breakdown of molecules which releases energy that is stored in ATP. Anabolism is the building of complex molecules using the energy stored in ATP. So, the ATP act as the cell's energy carrier, transferring energy from catabolism to anabolism.
As organisms live and grow, they're constantly in the process of making and breaking bonds in molecules. Metabolism is the sum of all the chemical reactions that take place in an organism. Metabolism includes catabolism and anabolism. Catabolism includes the processes that break down complex molecules into simpler molecules while harvesting their energy and storing it, usually in the form of ATP. Anabolism includes the processes that build more complex molecules from simpler molecules. The energy acquired through catabolic processes is used to drive anabolic processes. Neither catabolism nor anabolism is completely efficient, so at each step, some of the available energy is lost into the environment as heat.
And so then we move on to oxidation and reduction reaction. This is also known as the redox reaction. So the redox reaction is the transfer of electrons from an electron donor to an electron acceptor. Now, the electron that's donating is oxidized, and the one that's accepting the donation is the electron acceptor, which is being reduced. Now, this might seem backwards, so let me explain why. So electrons, they're already negative enough. So if the electron acceptors accept another electron, their overall electrical charge decreases as well. This is why they are being reduced. Now, electron donors are oxidized because in many reactions, their electrons are donated to oxygen atoms. And the reaction always occurs simultaneously, meaning it always happens at the same time. Cells use electron carriers to carry electrons, often in hydrogen atoms. And there's three important electron carriers. We first have nicotinamide adenine dinucleotide, or NAD+. Now, when NAD+ accepts electrons, it also picks up hydrogen atom, forming NADH. This carrier is crucial in cellular respiration, often in pathways like glycolysis and citric acid cycle, or the Krebs cycle. We also then have nicotinamide adenine dinucleotide phosphate, or NADP+. Now, similar to NAD+, NADP+ accepts electrons and forms NADPH, which is primarily used in anabolic pathways such as the Calvin cycle in photosynthesis to build molecules. Then finally, we have flavin adenine dinucleotide, or FAD. FAD can carry 2 electrons and 2 hydrogen atoms, forming FADH2. FADH2 plays an important role in the electron transport chain during cellular respiration. Now, these three electron carriers are going to be important later in the slides. So more information to that in a bit. So here's a figure of the redox reaction. So an electron donor donates their electron to an electron acceptor, and after that, they become oxidized. Now, the electron acceptor that accepted the electron becomes reduced because they gain the electron. And again, they become reduced because when they accept the electron right here, their overall electrical charge decreases because the electrons are negatively charged. Now, an acronym to remember this is the word oil rig. So OIL, meaning oxidation is loss, and RIG, meaning reduction is gained, referring to the electrons being lost and gained.
Many metabolic reactions involve the transfer of electrons from one molecule to another. This transfer of electrons involves two coupled reactions, oxidation and reduction. These are often called redox reactions. Cells use redox reactions to transfer electrons from one molecule to another within or between metabolic pathways, such as the Krebs cycle and electron transport chain. In an oxidation reaction, a molecule called the electron donor gives up one or more electrons to another molecule called the electron acceptor. In the oxidation reaction, the donor molecule becomes oxidized. A reduction reaction occurs when a molecule receives electrons from another molecule. The electron receptor therefore becomes reduced. The term reduced refers to the receptor's electrical charge. Because it's gained a negatively charged electron, the overall charge on this molecule becomes more negative and is said to be reduced. You can now see that the oxidation reaction must be coupled with a reduction reaction. In other words, if a molecule is to donate an electron and become oxidized, then another molecule must accept that electron and become reduced.
And so then we move on to ATP production and energy storage. So organisms release energy from nutrients, and this energy can be concentrated and stored in the form of high energy phosphate bonds, specifically in the molecule known as the adenosine triphosphate or ATP. Phosphorylation is the process by which an inorganic phosphate is added to a substrate. So essentially, it converts ADP to ADP, storing energy in the process. So cells phosphorylate adenosine diphosphate into ATP in 3 ways. We first have substrate level phosphorylation. So substrate level phosphorylation is the direct transfer of phosphate group from a molecule to ADP. We then have oxidative phosphorylation. Oxidative phosphorylation is when energy from respiration or redox reaction powers the addition of an inorganic phosphate to ADP. And then we have photophosphorylation. So photophosphorylation is when light energy drives the phosphorylation of ADP with inorganic phosphate during photosynthesis. So anabolic pathways use some energy of ATP by breaking a phosphate bond. So when ATP is broken down into ADP, we get energy. And energy is needed to convert ADP into ATP. So in summary, ATP and ADP function in a continuous cycle where ATP stores and provides energy for immediate cellular activities, and ADP is recycled into ATP through cellular processes, ensuring a constant supply of energy for the cell's need. And so then we move on to the role of enzymes in metabolism. So enzymes are organic catalysts, which means it increases the likelihood of a reaction.
Imagine a chemical reaction with the general form AB yields A plus B. In order for this reaction to occur, the reactant molecule, AB, must acquire enough energy to disrupt its stable electron configuration so that its electrons can rearrange to form A and B. This energy barrier is called the activation energy of the reaction. In living cells, enzymes serve as biological catalysts, speeding up reactions. Enzymes accomplish this task by lowering the activation energy of the reaction, making it easier for reactants to acquire the activation energy required to produce products.
Now, one thing you should take away before we dive into the enzyme classification based on reaction type is that enzyme always and most likely always will end with the suffix ase. Okay, now let's jump into the enzyme classification based on reaction type. We first have hydrolase. Hydrolase or hydrolysis is the breaking down of molecules by adding water. This is catabolic. An example of this is with lipase. Lipase breaks down lipid molecules. We then move on to isomerase. Isomerase is the arrangement of atoms within a molecule. It is neither catabolic nor anabolic. An example of this is with phosphoglucoisomerase, in which it converts glucose 6-phosphate into fructose 6-phosphate during glycolysis. We then move on to ligase or polymerase. This is when it joins two or more chemicals together, and this is anabolic. So acetyl-CoA synthetase is an example, in which it combines acetate and coenzyme A to form acetyl-CoA for the citric acid cycle. We then move on to lyase, in which it splits a chemical into smaller parts without using water. This is catabolic. An example of this is with fructose 1,6-biphosphate aldosase. It basically splits fructose 1,6-biphosphates into G3P and DHAP during glycolysis. We then have oxidoreductase. This is the transfer of electrons or hydrogen atom from one molecule to another. An example of this is with lactic acid dehydrogenase, and it oxidizes lactic acids to form pyruvic acid during fermentation. We then have transferase. It is when a moving functional group from one molecule to another, and this may be anabolic. An example of this is with hexokinase. It transfers phosphate from ATP to glucose in the first step of glycolysis. And so then we move on to the makeup of enzymes. Many enzyme or many protein enzymes are complete in themselves, meaning that they don't require additional molecules to catalyze reactions. However, there's an exception to this. Apoenzymes are inactive if not bound to non-protein cofactors. So cofactors are the non-protein substances that apoenzymes need to become active, and these include inorganic ions or coenzymes. Inorganic ions include metals such as iron, magnesium, zinc, or copper. And coenzymes are organic molecules that are either vitamins or anything that contain vitamins. And the binding of apoenzyme and its cofactor yields the name holoenzyme, which is the combination of apoenzyme and its cofactor. Not all enzymes are protein. Some RNA molecules are called ribozymes. So while most enzymes are protein, some enzymes are made out of RNA and called ribozymes. Ribozymes are RNA molecules that have the ability to catalyze specific biochemical reaction similar to protein-based enzymes. And so then we move on to the makeup of a protein holoenzyme. So again, the apoenzymes are inactive if not bound to non-protein cofactors. So in this case, we have an inorganic cofactor and a coenzyme cofactor, which makes up the active site. Now, this whole structure is also known as the holoenzyme. So every structure here, once it's made, it's known as the holoenzyme. And so then, this is the effect of enzyme on chemical reactions. Now, I already played a video on this earlier, so I'll just be explaining it here. So enzymes catalyze the reaction by lowering the activation energy. That is the energy needing to trigger the reaction. So this is without an enzyme, and this is with an enzyme. And so then we move on to the enzymes fitted to substrate. So let's look at the key concepts. We first have the substrate. The substrate is the molecule that acts upon the enzyme. And then we have the enzyme active site. The enzyme has an active site shown as the blue shape. The active site has a structure that is similar to the substrate shape, but not an exact match at first. So then we go into the induced fit model. The enzyme and substrate interaction follows the induced fit model. Initially, the enzyme's active site is complementary, but not the exact fit for the substrate. So when the substrate binds, it causes the enzyme active site to undergo a slight conformational change, which adjusts to fit the substrate more closely. And this allows the enzyme to better catalyze the reaction. So in other words, this picture here shows the induced fit model of an enzyme-substrate interaction. An enzyme active site is generally complementary to the shape of its substrate, but a perfect fit between them does not occur until the substrate and enzyme bind to form a complex, at which time the enzyme active site changes to fit the substrate more closely. And so let's look at the process of the enzymatic activity. So we first have the enzyme binding to a substrate, which is in this case the fructose 1,6 biphosphate. So the enzyme fructose 1,6 biphosphate aldolase associates with the substrate molecule fructose 1,6 biphosphate at its active site right here. The substrate has two phosphate groups at the end of each molecule. 2. The formation of enzyme substrate complex. So once bound, an enzyme substrate complex forms. The binding causes a slight change in the enzyme's active site to fit the substrate more closely. This is the induced fit. So as you can see, this was kind of like round shape, and now it is the exact same fit as the substrate. Step 3, catalysis, the breaking of bonds. So the enzymes induces the breaking of bonds within the substrate, splitting fructose 1,6 biphosphate into two 3-carbon molecules, which is the glycerhalide 3-phosphate or G3P and dihydroxyacetone phosphate or DHAP. So then we get the formation of products. The products of the reaction are G3P right here and DHAP, both 3-carbon molecules with phosphate groups attached. These products are then released from the enzyme active site. And then finally we get to the last step. The enzyme is ready for another reaction. So after the reaction, the enzyme releases the products and then returns to its original shape. It is now ready to bind to another substrate molecule and repeat the process.
Although every enzyme performs a different reaction on different substrates, they all go through the same general steps in accomplishing their reaction. These general steps include substrate binding, formation of the enzyme-substrate complex, product formation and dissociation, and enzyme recovery. Like all catalysts, enzymes are unchanged by the overall reaction. Now let's look at each step in more detail. The first step in any enzymatic reaction is the binding of the substrate by the enzyme. The enzyme includes an active site that can accommodate the size, shape, and electron configuration of its substrate or substrates. According to the induced fit model of enzyme-substrate interaction, a perfect fit does not form until the enzyme and substrate bind to form an enzyme-substrate complex. After the substrate is bound in the active site, the enzyme-substrate complex is formed. The shape of the enzyme may change to allow for an efficient chemical reaction, and amino acids may accept or donate protons or electrons to facilitate the chemical reaction. Once the enzyme-substrate complex is formed, and any reactive groups have moved to the right position, the reaction proceeds and products are formed. The products dissociate rapidly, leaving an empty active site. After the products leave the active site, the enzyme returns to its original configuration. Any groups that donated or accepted protons or electrons are also returned to their original state. Like all catalysts, the enzyme is unchanged by the overall reaction.
And so we look at the enzyme activity. Many factors influence the rate of enzymatic reactions, starting off with temperature. So enzymes have an optimal temperature range in which they function most effectively, and in this case, they function most effectively at 37 Celsius, or in Fahrenheit, it's 98.6. If the temperature is too low, enzyme activity slows down because molecular movement is reduced. If the temperature is too high, the enzyme may denature or lose its function, leading to loss of function. We then have pH. Each enzyme has an optimal pH range. This is around 7.2. Deviations from this range can affect the shape of the enzyme's active site or the substrate, reducing the enzyme's effectiveness or causing it to denature. Denature meaning again to lose its structure. We then also have the enzyme and substrate concentration. So the concentration of enzyme and substrates impact the reaction rate. If there are more substrate than available enzymes, the reaction will proceed slower until enzyme availability increases. If the enzyme concentration is too high, reaction will proceed faster as long as there are enough substrates available. And finally, we have the presence of inhibitors. So inhibitors are molecules that bind to enzymes and reduce its activity. They can be competitive inhibitor or non-competitive inhibitor. So here's a representative effect of temperature. So again, enzymes have an optimal temperature range, which is around 37 degrees Celsius. Rising temperature enhances enzymatic activity to a point, but above some optimal temperature range, an enzyme denatures and loses its function. And this image here shows the denaturation of protein enzymes. So the picture in the left right here shows a functional protein. In its natural state, the protein or the enzyme has a specific three-dimensional shape. This structure is held together by non-covalent bonds such as hydrogen bonds, ionic bonds, and van der Waals forces. The protein shape is essential for its function, particularly in active site, where it binds to substrates and catalyzes reaction. Now, the image in the right here shows a denatured protein. So when exposed to extreme conditions like high temperature, drastic pH changes, or harsh chemicals, the non-covalent bonds that stabilize the protein structure begin to break. As these bonds break, the protein loses its secondary and tertiary structures, which is the complex folding patterns that allow the protein to function. This results in the protein unfolding into a disordered linear shape. And once denatured, the protein is no longer in its proper shape, and the active site is lost, meaning that the enzyme can no longer bind to substrate to catalyze reactions. The enzyme is now non-functional. And then we look at the representative effect on pH and substrate concentration on enzyme activity, starting off with the pH. So enzymes typically have a optimal pH, at which point enzyme activity reaches a maximum. And the enzyme's optimal pH is around 7.2. Then we look at the substrate concentration. At low substrate concentration, enzyme activity rises as the substrate concentration increases, with more active sites becoming engaged. And when all the active sites are occupied, this is known as the saturation point, in which the enzyme activity reaches its peak, and the further increase in substrate do not affect the enzyme activity. And so then we move on to the control of enzymatic activity. So enzymes can be regulated / controlled to ensure that they are active only when needed. And one way is through activators. So some enzymes are activated when cofactor binds to a site other than the active site. So in other words, when a cofactor binds to an allosteric site, it causes a conformational change in the enzyme structure. This change in the shape can alter the enzyme's active site in such a way that it becomes properly shaped to bind the substrate, activating the enzyme. So this image here shows an allosteric activation. So we first look at the distorted non-functional active site. So initially, the enzyme active site is distorted and unable to bind to substrate. In this state, the enzyme is inactive and cannot catalyze a reaction. So let's look at the allosteric site. The enzyme has an allosteric site which is separate from the active site. Sorry. So when an allosteric activator, which is represented in the green molecule right here, binds to this site, it triggers a change in the enzyme shape. The binding of the activator at the allosteric site causes the enzyme to undergo a conformational change. This change alters the shape of the enzyme, specifically the active site, making it functional. After the active site binds, the enzyme active site becomes properly shaped, allowing the substrate, or the purple molecule here, to bind to the active site effectively. This activates the enzyme, allowing it to catalyze reaction. And then we move on to inhibitors. Inhibitors are substances that block enzyme activity by interfering with the enzyme's ability to catalyze reaction. So again, inhibitors, substances that block an enzyme's activity. And this includes competitive inhibitor and non-competitive inhibitor. So with competitive inhibitor, they directly compete with substrates for an enzyme active site. Inhibitors can bind permanently or reversibly. And if reversibly, this inhibition can be overcome by increasing the concentration of the substrate. With non-competitive inhibitor, it binds to an allosteric site, causing a change in the enzyme shape that blocks or reduces its activity. This type of inhibition is not affected by the concentration of the substrate. So adding more substrate doesn't reverse the inhibition. Feedback inhibition controls the action of some enzymes. So, in other words, the feedback inhibition is a precise control mechanism in which the end product of a metabolic pathway acts as an inhibitor to an enzyme earlier in the pathway, ensuring that the pathway is regulated efficiently based on the cell's current need. So let's look at the competitive inhibition of enzyme activity. So figure A shows an inhibitory molecule, which are similar in shape to substrate molecules that compete for and block active sites. Reversible inhibition can be overcome by an increase in substrate concentration.
A competitive inhibitor is a substance that is structurally similar to the enzyme substrate, but which does not react to form products. For example, sulfanilamide is a competitive inhibitor for the enzyme whose normal substrate is para-aminobenzoic acid, PABA. Note the similarity in molecular structure of PABA and sulfanilamide. In competitive inhibition, competitive inhibitors compete with the substrate molecules for the binding site, slowing catalysis. However, if the concentration of substrate is much higher than the concentration of competitive inhibitor, the substrate will outcompete the inhibitor for the enzyme active site, and catalysis will continue.
Then we look at non-competitive inhibition at an allosteric site. So the allosteric site. Enzymes have an additional site known as the allosteric site, which is separate from the active site, where substrates normally bind. In non-competitive inhibition, an allosteric inhibitor binds to the enzyme at the allosteric site rather than the active site. And when the allosteric inhibitor binds, it causes a conformational change in the enzyme. And this shape change distorts the active site, either reducing or completely stopping the enzyme's ability to catalyze the reaction, even if a substrate is present and can still bind. So unlike competitive inhibitors, non-competitive inhibitors do not compete with the substrate for the active site. Instead, they change the shapes of the enzyme so that it can no longer function properly regardless of substrate concentration.
Non-competitive inhibitors bind to a location on the enzyme other than the active site. For example, the HIV medication nevirapine is a non-competitive inhibitor of HIV reverse transcriptase. Nevirapine does not bind to the reverse transcriptase active site. The binding of the inhibitor causes the enzyme to change shape, so the active site is no longer able to perform the reaction efficiently. Since the non-competitive inhibitor binds in a different binding site than the substrate, increasing the concentration of substrate has no effect on non-competitive inhibition.
And so we look at feedback inhibition, starting off with the pathway operates. So at the start of the pathway, a substrate binds to an active site of enzyme 1, initiating the process. The substrate is converted through a series of steps producing intermediates, which is the intermediates A and B, as each enzyme in the pathway carries out its function. And eventually, the end product is formed at the final step of the pathway. Once enough end product is produced, it acts as an allosteric inhibitor. The end product binds to an allosteric site on enzyme 1, causing a conformational change in the enzyme structure. This changes the shape of the enzyme, making the active site non-functional. With the end product bound to enzyme 1, the pathway is effectively shut down because the substrate can no longer bind to enzyme 1. This prevents further production of intermediates and the end product. The inhibition of the first enzyme stops the entire pathway, ensuring that energy and resources are not wasted producing excess end products. And so then we get to the resumption of pathway. When the levels of end product decrease, the inhibition is lifted and the pathway can resume. So we then look at the importance of the feedback inhibition. So the feedback inhibition is a regulatory mechanism that allows cells to control the flow of metabolic pathways efficiently. By inhibiting the first enzymes in the pathway, the cell can prevent the overproduction of substances, conserving energy and resources. This type of inhibition is reversible, meaning that pathway can restart when the concentration of the end product decreases. So in summary, this diagram shows how the end product of a metabolic pathway inhibits the first enzyme in the pathway, effectively shutting down the process through a feedback inhibition to maintain balance and prevent waste. And then we move to carbohydrate catabolism. So many organisms oxidize carbohydrates as primary energy source for anabolic reactions. Glucose is the most common carbohydrate used, and glucose is catabolized by two processes, which are cellular respiration and fermentation. So with cellular respiration, that is a process in which results in the complete breakdown of glucose to CO2 and H2O. With fermentation, this results in an organic waste product. So here's a summary of glucose catabolism. So glycolysis, or the EMP pathway. Glycolysis is a starting point for both respiration and fermentation. It involves the breakdown of glucose into two molecules of pyruvic acid. Along with the pyruvic acid, glycolysis produces two molecules of ATP and two molecules of NADH. These are the electron carriers. This process occurs in the cytoplasm of the cell and does not require oxygen. So starting off with the first pathway, which is respiration. So after glycolysis, in the presence of oxygen, the pyruvic acid is converted into acetyl-CoA, which enters the citric acid cycle, or the Krebs cycle. In the citric acid cycle, pyruvic acid is fully oxidized into carbon dioxide, and more electron carriers like NADH and FADH2 are produced along with a small amount of ATP. The NADH and FADH2 produced during the citric acid cycle transfer their high energy electron to the electron transport chain, or the ETC. In the ETC, electrons are passed through a series of proteins, and their energy is used to generate a large amount of ATP. At the end of the ETC, or the electron transport chain, oxygen acts as a final acceptor, combining with electrons and hydrogen ions to form water. This pathway results in the production of significantly more ATP compared to fermentation, making it more efficient energy generating process. And so then we move on to fermentation, or pathway number 2. So if oxygen is absent or unavailable, pyruvic acid undergoes fermentation rather than respiration. In fermentation, pyruvic acid is converted into organic end products such as lactic acid or ethanol, depending on the type of organism. Unlike respiration, fermentation does not involve the citric acid cycle or the electron transport chain. So as a result, fermentation produces much less ATP and leads to incomplete oxidation of pyruvic acid. So NADH produced in glycolysis is used to regenerate NAD+, ensuring that glycolysis can continue in the absence of oxygen. And so then we move on to glycolysis. Glycolysis is commonly the first step in catabolism of glucose via both respiration and fermentation. It occurs in cytoplasm of most cells, and it involves a splitting of 6-carbon glucose into 2 3-carbon sugar molecules. So let's break that down, starting off with the glucose molecular form. So the glucose molecular form has 6 carbons, 12 hydrogens, and 6 oxygen. So when it says it involves a splitting of 6-carbon glucose into 2 3-carbon sugar molecules, it's referring to the carbon in the glucose splitting into 2 3-carbon sugar molecules. And this becomes pyruvate, or the pyruvic acid. Glycolysis also involves a type of ATP production known as the substrate level phosphorylation, which is the direct transfer of phosphate between two substrates, meaning it converts ADP into ATP. So ADP meaning adenosine diphosphate, di meaning 2, and converting it into adenosine triphosphate, tri meaning 3. So essentially, it's gonna add another phosphorus in here, or sorry, another phosphate.
The metabolism of glucose is of central importance to all living organisms and is in fact performed in almost every cell. As one of the major sources of energy in living systems, glucose is made and broken down constantly in living cells. The metabolic pathway for the breakdown of glucose is called glycolysis, from the Greek glucose for sweet or sugar and lysine for breaking apart. Glycolysis is sometimes called the Embden-Meyerhof pathway after its discoverers. Glycolysis consists of two main phases, the preparatory stage and the energy conservation stage. The preparatory stage uses 2 molecules of ATP to phosphorylate glucose, making it easier to break it apart into 2 3-carbon molecules. In the energy conservation stage, 4 ATP and 2 NADH molecules are produced, along with 2 pyruvic acid molecules. At the end of glycolysis, the energy found in glucose has been converted to a net of 2 ATP molecules and 2 NADH molecules.
And so then we look at glycolysis by the EMP pathway. We start with the energy investment stage, or step 1, 2, 3. Step 1. Glucose, right here, is phosphorylated by ATP to form glucose 6-phosphate. Steps 2 and 3. Glucose 6-phosphate is rearranged to form fructose 1,6-phosphate, which is phosphorylated by another ATP molecule to become fructose 1,6-biphosphate. Okay, second stage, lysis stage, or the lysis stage, sorry. This contains steps 4 and 5. So step 4, the fructose 1,6-biphosphate is split into two 3-carbon molecules, which are the glyceraldehyde 3-phosphate, or G3P, and dihydroacetone phosphate, or DHAP. And step 5 is when the DHAP over here is rearranged to form another G3P. So now there's two G3P molecules. And then we move on to the last stage, which is the energy conserving stage. This stage contains the last few remaining stages left. So step 6. Each G3P is phosphorylated and two NAD plus molecules are reduced to form NADH. Step 7. Two ADP are phosphorylated by substrate level phosphorylation to form two ATP molecules. Steps 8 and 9. A water molecule is removed from each substrate as 3-phosphoglyceric acid is converted into phosphoenol pyruvic acid, or the PEP. And then finally, the last step, step 10. Two ADP are phosphorylated again by substrate level phosphorylation to form two more ATP. And the final product is the pyruvic acid. So the final product of glycolysis is two pyruvic acids molecule, two NADH molecules, and it had a net gain of two ATP. So while there are four ATP molecules that were produced in total, two ATP were used in the energy investment stage. So the net result is two ATP. So this image here shows the example of substrate level phosphorylation. So the diagram illustrates the conversion of PEP, right here, to pyruvic acid, while transferring a phosphate group of ADP to produce ATP. This is the phosphorylation. And this process occurs during glycolysis and is a crucial way to generate ATP, especially under conditions where oxygen is unavailable or limited. And then we move on to cellular respiration. Cellular respiration is a metabolic process that involves the complete oxidation of substrate molecules and production of ATP via a series of redox reactions. So again, it is the resultant of pyruvic acid completely oxidized to produce ATP by a series of redox reactions. And there's three stages of cellular respiration, starting off with the synthesis of acetyl-CoA. So the pyruvic acid produced from glycolysis is converted into acetyl-CoA. This is the first preparatory step for the citric acid cycle. In this process, a carbon atom is removed from the pyruvic acid as CO2, and the remaining two carbon molecules bind to the coenzyme, which makes up the CoA, forming acetyl-CoA. NADH is also produced during this step, which will later be used in the electron transport chain, or the ETC. The second step is the Krebs cycle. Acetyl-CoA enters the Krebs cycle, where it is further oxidized. During this cycle, ATP, NADH, FADH2, and CO2 are produced. NADH and FADH2 are crucial for transferring electrons to the electron transport chain. And then finally, we get to this final series of redox reaction. The electron transport chain, or the ETC, is the final stage of cellular respiration, where most of the ATP is generated. So the NADH and FADH2 from Krebs cycle donate their electrons to ETC, where a series of proteins transfer these electrons and pump protons across the mitochondrial membrane. The flow of proton back to the ATP synthase generates ATP, a process known as the oxidative phosphorylation. Oxygen serves as the final electron acceptor, combining with the electron and protons to form water. So let's get into the citric acid cycle, or the Krebs cycle. Okay. So we first have a acetyl-CoA, a 2-carbon molecule that combines with a oxoacetic acid, which is a 4-carbon molecule, to form citric acid, a 6-carbon molecule. The citric acid then undergoes a structural change to form isocitric acid, which is still a 6-carbon molecule. The isocitric acid is oxidized, reducing NAD plus to NADH, and producing a ketoglutaric acid, a 5-carbon molecule, and CO2. This is the first release of carbon dioxide. The a-ketoglutaric acid is further oxidized, reducing NAD plus to NADH, and producing succinyl-CoA, a 4-carbon molecule, and another molecule of CO2. So then the succinyl-CoA then converts into succinic acid through a substrate-level phosphorylation. The release of CoA from succinyl-CoA provides the energy for this reaction. This energy is used to convert GDP to GTP, or in some cases, ADP to ATP, depending on the specific cell type. The succinic acid is then oxidized into fumaric acid, reducing FAD into FADH2, another electron carrier. Water is then added to fumaric acid, producing malic acid. The malic acid is oxidized, reducing NAD plus into NADH, and regenerating oxaloacetic acid to begin the cycle once again. So the final product of one turn cycle are 3 NADH, one right here, one right here, one right here, one FADH2 right here, one ATP or GTP right here, 2 CO2 right here, and one oxaloacetic acid right here.
The Krebs cycle, or citric acid cycle, is one of the 3 stages of cellular respiration. During respiration, cells completely oxidize metabolites to carbon dioxide and water to obtain chemical energy. However, pyruvic acid generated by glycolysis cannot enter the Krebs cycle directly. First, it must be decarboxylated in a step that's often called the bridge or transition step. During the bridge step, one carbon atom is removed from pyruvic acid, forming acetate. It is then attached to coenzyme A, resulting in a complex called acetyl-CoA. Acetyl-CoA can then enter the Krebs cycle. The Krebs cycle is a carefully choreographed process that efficiently extracts the energy in the pyruvate molecule in 8 enzymatic steps. The energy is released in the form of the electron carriers NADH and FADH2 and the energy carrier guanosine triphosphate, GTP. Ultimately, during one pass through the bridge step and Krebs cycle, enough energy is extracted from a single molecule of pyruvic acid to generate 15 molecules of ATP.
And then we move on to the electron transport chain.
Many organisms utilize systems called electron transport chains to generate usable energy in the form of ATP. Electron transport chains consist of a series of membrane-bound proteins contained in the plasma membranes of prokaryotes and the inner membranes of mitochondria of eukaryotes. As electrons are passed along the chain, the energy released is used to pump protons across the membrane. This active transport produces a proton gradient, which can be used to generate usable energy through a process called chemiosmosis. It's helpful to think of a proton gradient as water held behind a dam. Much as dams can drive turbines and generate electricity by allowing water to flow from a high area to a low area, cells can generate usable energy by allowing protons to flow from areas of high concentration to areas of low concentration. This diffusion of protons across the membrane provides the power for ATP synthase to generate ATP.
And then finally, we get into electron transport. So there's 2 types. There's aerobic respiration and anaerobic respiration. So aerobic respiration is when oxygen serves as the final electron acceptor, whereas anaerobic respiration is when the molecule other than oxygen serves as the final electron acceptor. So aerobic respiration, this type of respiration occurs when oxygen is available, and it is the most efficient way for cells to produce ATP. Anaerobic respiration is when the environment lacks oxygen, and this typically generates less ATP compared to aerobic respiration. And then we move on to the electron transport chain molecules. Now, I'll play a video here explaining it since I think the guy explained it really well.
Recall that electrons are carried to the electron transport chain from glycolysis, the bridge step, and the Krebs cycle by the electron carriers NADH and FADH2. In prokaryotes, all 3 of these processes occur in the cytoplasm. The electron transport chain of prokaryotes is located on the cytoplasmic membrane. First, let's look at the components of the electron transport chain, and then we will see how it's used to produce ATP. Flavoproteins are integral membrane proteins which often contain the coenzymes flavin or flavin adenine dinucleotide, FAD. Electrons from NADH usually enter the electron transport chain through a large protein complex that contains a flavin called flavin mononucleotide or FMN. This complex also contains clusters of iron and sulfur in which the iron can alternate between the reduced and oxidized states. Ubiquinones, such as coenzyme Q, are small lipid-soluble non-protein electron carriers. Ubiquinones act as shuttles between different portions of electron transport chains, ferrying electrons from one part of the membrane to another. Here, coenzyme Q is reduced by the FMN complex and oxidized by the cytochrome B cytochrome C1 complex and can therefore carry electrons between them. Cytochromes are membrane-bound proteins, which contain a heme group, the same iron-containing molecule found in the hemoglobin of blood. This electron transport chain contains 3 cytochrome complexes made up of 5 cytochromes: cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Electrons are passed one at a time sequentially from one of these cytochromes to the next one until it reaches the cytochrome A3 complex. Next to the cytochrome complex is ATP synthase. This is a protein channel through which protons diffuse. A portion of this channel acts as an enzyme to catalyze the formation of ATP. Now let's see how these electron transport chain components work together to convert potential energy carried in electron carriers to ATP. Electrons carried by NADH enter electron transport chains through flavoproteins. NADH passes a hydrogen ion with 2 electrons to an electron acceptor, such as FMN, oxidizing NADH to NAD and reducing FMN, which picks up another proton from the surrounding medium to form FMNH2. These 2 protons are pumped to the outside of the membrane. The electrons are then passed from FMNH2 to coenzyme Q, reducing it. Coenzyme Q becomes oxidized again when it gives up its electrons to the cytochrome B cytochrome C1 complex. Electrons carried by FADH2 enter the electron transport chain at coenzyme Q rather than FMN. Because these electrons enter at a lower energy level, this will result in a lower yield of ATP per FADH2 than that produced with NADH. Electrons are passed one at a time from coenzyme Q to the cytochrome B cytochrome C1 complex, which may also serve to pump protons to the outside of the membrane. The electrons are then passed to cytochrome C, which delivers them to the cytochrome A3 complex. This complex serves as a proton pump, but also serves to pass the electrons off to the final electron acceptor. In mitochondria and aerobic bacteria, the final electron acceptor is oxygen, which is reduced to form water. In aerobic respiration, the final electron acceptor is molecular oxygen, O2. In anaerobic respiration, the final electron acceptor is a substance other than oxygen, such as nitrate ion, sulfate ion, or carbonate ion. Electron transport chains are used to generate a proton gradient, which is a buildup of protons on one side of a membrane. This proton gradient is an electrochemical gradient, since there's a gradient of concentration and of charges across the membrane. It can also be considered a pH gradient, since pH represents proton concentration. In respiration, the protons diffuse across the membrane through protein channels called ATP synthases. As the protons diffuse through ATP synthase, the energy of the proton gradient is used to synthesize ATP from ADP and phosphate.
So here's a summary of the video. NADH and FADH2 donate their electrons to the electron transport chain. These electrons move through a series of membrane proteins, and they lose energy in the process. Oxygen serves as the final electron acceptor, forming water. And then finally, the proton gradient drives ATP synthase, which generates ATP through oxidative phosphorylation, the primary method of ATP production during cellular respiration. So this mechanism allows the cell to produce a large amount of ATP efficiently. So here's the summary of an ideal prokaryotic aerobic respiration of one molecule of glucose. Although there are 40 ATP produced in here, 2 of them were used from glycolysis. Now, if you recall, we invested 2 ATPs to make a net gain of 2 more ATPs. So if you add up every pathway, we get 38 in total as our net gain. We also get the NADH. 10 were produced, and only 2 FADH2 were produced. And then we move on to fermentation. So we already went over respiration, so now it's fermentation's turn. So this whole image here demonstrates an example of fermentation, which is the process that occurs when cells convert pyruvic acid, which are produced from glycolysis, into various organic products in the absence of oxygen. So this figure here shows 2 key pathways. 1. Lactic acid formation. So the pyruvic acid here is reduced by NADH to form lactic acid. The NADH is oxidized back into NAD+, which can then be reused in glycolysis to continue generating ATP. This process is seen in certain bacteria and in muscle cells when oxygen is limited. So then we go into alcohol fermentation. So the pyruvic acid is first converted into acetaldehyde right here by the release of carbon dioxide. Acetaldehyde is then reduced by NADH to form ethanol. NADH is again oxidized to NAD+, allowing glycolysis to continue. So these fermentation allows cells to regenerate NAD+, which is essential for glycolysis to proceed and continue producing small amounts of ATP under anaerobic conditions, which is the absence of oxygen. So this slide right here shows the comparison of aerobic respiration, anaerobic respiration, and fermentation. We start off with aerobic respiration. In aerobic respiration, oxygen is required. The type of phosphorylation is substrate level and oxidative, and the final electron acceptor is oxygen. Now, the potential molecules of ATP produced per molecule of glucose is 38 in prokaryotes and 36 in eukaryotes. Then we move on to anaerobic respiration. Anaerobic respiration does not require oxygen, and it has the same type of phosphorylation as aerobic respiration, which is substrate level and oxidative. Now, the final electron acceptor for anaerobic respiration is nitrate, sulfate, carbon dioxide, or any external acquired organic molecules. The potential molecules of ATP produced per molecules of glucose in anaerobic respiration is 4 through 36. Then we go to fermentation. Fermentation does not require oxygen. Now, the type of phosphorylation in fermentation is substrate level. The final electron acceptor is the cellular organic molecules, and the potential molecules of ATP produced per molecules of glucose in fermentation is 2. So this comparison shows that aerobic respiration is the most efficient process for ATP production. While fermentation yields the ATP, it primarily serves as the backup energy generation method when oxygen is unavailable. We then move on to the representative fermentation products and the organisms that produce them. So here are the fermentation processes in different organisms and the product that they generate. We start off with the Propionibacterium. Propionibacterium produces carbon dioxide and the propionic acid. And the end product example is Swiss cheese. We then have Aspergillus, Lactobacillus, and the Streptococcus. This fermentates and gets lactic acid. Now, the end product examples are cheddar, cheese, yogurt, and soy sauce. We then move on to Saccharomyces. When it fermentates, it produces carbon dioxide and ethanol, and this makes wine and beer. Then we have Clostridium. When it fermentates, it makes acetone and isopropanol. So the end product examples here are nail polish remover and rubbing alcohol. So these organisms undergo fermentation to regenerate NAD plus from NADH, allowing glycolysis to continue in the absence of oxygen. These specific fermentation pathways vary depending on the organism and result in different products that are utilized in various industrial and food applications. So we then go on to other catabolic pathways. Lipid and proteins contain energy and their chemical bonds. It is similar to carbohydrates. It can be converted into precursor metabolites. So by that bullet point, it's saying that during their breakdown, the component of lipid and proteins can be transformed into intermediate molecules, which are crucial for other cellular processes. It also serves as substrate in glycolysis and Krebs cycle, meaning that the breakdown of products of lipid and proteins can enter metabolic pathways such as glycolysis and the Krebs cycle to generate ATP, which is used for energy for the cell. This means that cell can use not just glucose, but also lipid and proteins to produce energy through these pathways. Protein catabolism. So, proteins are made of long chains of amino acids called polypeptides, with different R groups representing different side chains for each amino acid. There are enzymes that break down polypeptides into individual amino acids. This process occurs outside the cell in the extracellular fluid with the help of water molecule, which is the hydrolysis. Once broken down into individual amino acids, they enter the cytoplasms for further processing. Inside the cell, the amino group NH2 is removed from the amino acid in a process called deamination, right here. This produces ammonia, or NH3, which is usually converted into a less toxic substance, such as urea in humans, and excreted. The remaining part of the amino acid after the removal of the amino group is called the carbon skeleton. It is then processed to enter the citric acid cycle, or the Krebs cycle, where it can be used to produce ATP for the cell. So this process allows the proteins to be used as a source of energy when necessary, such as during periods of fasting or low carbohydrates intake. And so then we move on to photosynthesis. Many organisms synthesize their own organic molecules from inorganic carbon dioxide. Most of these organisms capture light energy and use it to synthesize carbohydrates from CO2 and H2O by a process called photosynthesis. So chemical and structures. Chlorophylls. Chlorophylls are important to organisms that capture light energy with pigment molecules. Chlorophylls are composed of hydrocarbon tail attached to a light-absorbing active site centered on magnesium ion. Active sites are structurally similar to cytochrome molecules in ETC. The structural differences cause absorption at different wavelengths. So, photosystems. Photosystems. The photosystem is the arrangement of molecules of chlorophyll and other pigments to form light-harvesting matrices. It is embedded in a cellular membrane called the thylakoid. So in photosynthetic cells, the thylakoid is the portion of cellular membrane containing the light-harvesting photosystems. In prokaryotes, thylakoids are found as infoldings of the cytoplasmic membrane. In eukaryotes, referring to plant and algae, the thylakoids are formed from the inner membrane of the chloroplast and are organized in stacks called grana. Stroma is the space between the outer membrane of grana and thylakoid membrane. So essentially, the stroma is the fluid space surrounding the grana and thylakoid membrane inside the chloroplast. The stroma is the site of the light-independent reaction of photosynthesis. So here's an image of a photosynthetic structure in a prokaryote. So image A. Image A shows a photosystem and chlorophyll in the thylakoid membrane. So we look at the chlorophyll. The chlorophyll molecules are responsible for capturing light energy that are embedded within the thylakoid membrane. The thylakoid membrane, this is where the light reactions of photosynthesis occur. In prokaryotes, it is part of the cytoplasmic membrane folded to form the compartments called thylakoids. The photosystems. The photosystem is a group of chlorophyll molecules and associated proteins that capture light energy. And then we look at the chlorophyll structure. The chlorophyll structure includes a long hydrocarbon tail that anchors into the thylakoid membrane and a central active site with magnesium ion, which helps absorb light energy. Then image B shows a transmission electron micrograph of a cyanobacterium. This TEM shows the actual structure of thylakoid membranes in a cyanobacterium, a type of prokaryotic organism in which the membrane is folded into layers to increase the surface area for photosynthesis. Integration and regulation of metabolic functions. So, cells synthesize or degrade channels and transport proteins. This means that cells can adjust their transport mechanisms by synthesizing or degrading proteins that allow substances to enter or exit the cell based on metabolic needs. Cells often synthesize enzymes only when the substrate is available. This means that enzymes are produced in response to the presence of their substrate, conserving resources by not producing enzymes unnecessarily. Cells catabolize the more energy-efficient choice if two energy sources are available. This one's kind of self-explanatory. It's saying that when multiple energy sources are available, the cell prioritizes the more efficient one for energy production. Cells synthesize metabolites they need and cease synthesis if the metabolite is available. This means that cells regulate metabolite synthesis based on availability, preventing wasteful production of compounds that are already present. Eukaryotic cells isolate enzymes of different metabolic pathways within membrane-bound organelles. So in eukaryotic cells, enzymes for different pathways are compartmentalized in organelles like the mitochondria or the endoplasmic reticulum, ensuring efficient regulation and separation of processes. Cells use allosteric sites on enzymes to control activity. So this means that cells regulate enzyme function by using allosteric sites, where molecules bind to either activate or inhibit the enzyme activity without binding to the active site. And finally, we move on to the feedback inhibition, in which slows/stop anabolic pathways when product is abundant. So when sufficient product has been made, feedback inhibition prevents the overproduction of substances by inhibiting enzymes in anabolic pathways, conserving energy and resources. Metabolism, the big picture.
Now that you've learned about different catabolic and anabolic processes of microorganisms, let's see how these fit together into a complete picture. Proteins can be broken down by protease enzymes into amino acids, which can be further broken down through the glycolysis and Krebs cycle pathways. Nucleic acids can be broken down by nuclease enzymes into nucleotides that can also enter the glycolysis pathway. Polysaccharides and other sugars can be converted into glucose and further broken down through glycolysis and the Krebs cycle. Lipase enzymes break lipids into glycerol, which enters the glycolytic pathway, and into fatty acids, which enter just prior to the Krebs cycle. Fermentation is also considered catabolism since it converts pyruvic acid into other 2 and 3 carbon molecules, as well as carbon dioxide. Recall that anabolism is synthesis of complex molecules from simple molecules. Essentially, anabolism is the reverse of catabolism. Note that synthesis of proteins, nucleic acids, polysaccharides, and lipids is the reverse of their catabolism. The pentose phosphate pathway uses an intermediate from glycolysis to produce 5-carbon sugars called pentoses. These pentoses are then useful in synthesis of nucleotides. Photosynthesis is primarily an anabolic reaction, using light as an energy source to build complex molecules like glucose from carbon dioxide.
And that should be it with the video. Good luck on the exam, guys.